Hi everyone. So first I would like to introduce myself as my name is Dr. Vijay Bhaskar Reddy, Associate Professor, Department of Chemistry, H&S Department. I'm handling environmental science subject. So today we are going to discuss about the bioremediation. So what is the bioremediation? What are the types are there? What are the process will be involved? What are the strategies to control the bioremediation? So these are all keywords or topics we discussed in this lecture. So first one is what is the bioremediation and the types of bioremediation is the classification of the bioremediation, either in situ bioremediation or ex situ bioremediation. What is the in situ, what is the ex situ, what is the advantages and disadvantages of both, and the comparison we discussed here. And then what are the process will be involved in the bioremediation, and what are the strategies to control the bioremediation. So these are all topics we discussed in this lecture. Next, first one is the bioremediation. So first, what is the bioremediation? In the name itself is the, so bio means biology, remediation means to solve a problem. So whatever problem in that environment, either water, the contaminants in the either water, soil or atmosphere, so that problem will be solved by using the some of the biological related, either microorganisms or bacteria or microbes or either plants, whatever that will be used, and solve that contaminants will be removed from the site is called the bioremediation. So now could simply say to use biological organisms. So there are different types of biological organisms are there, either bacteria, algae, fungi, microbes, different types of microbes, and plant materials, plant extracts. These are all will be using and to solve environmental problem, that is called the bioremediate. So whatever that will be used is called the bioremediate. That process is called the bioremediation. Now coming to the bioremediation is the process by using the microorganisms to remove environmental pollutants. So whatever the environmental pollutants present in the either soil or water or air or toxic some of the compounds or materials found in the environment, that will be removed from that point by using the biological microorganisms is called the bioremediation. So here in this one, microbes, so whatever microorganisms we are using for the bioremediation, that will be working or serves as a scavengers. Scavengers means simply say decomposers. So these are the very good decomposers. So decomposers means a degradation of the organic matter or organic compounds to the simpler compounds or very low molecular weight compounds. So here that is the scavengers. So the removal of organic waste by microbes for environmental cleanup in the essence of bioremediation. So means here the microbes will be used to remove the contaminants from the environment of either soil or water or air. Now we can go the what are the bioremediation is a triple corners process. So here there are three key elements will be present in that one. First one is the pollutants, second one is the environment, third one is the organism. So means that's why this is the triple corner process, the bioremediation. First, what type of pollutants will be available in that environment? Either inorganic pollutants like salts or metals or materials or metal salts, heavy metals are mixed with the metals. So these are all coming under the inorganic pollutants. Organic pollutants means is the carbon, hydrogen, heavy molecular weight compounds that will be classified as a solids, liquids and gases. So that is the organic, and the pollutants either inorganic or organic or heavy materials also. Now coming to the environment. So where that pollutants will be generally in the environment, we can remove it in the soil or water or air. What type of organisms will be generally used for the bioremediation? So microorganisms, plants, plant extracts or enzymes. So these are all will be used as a organisms or microorganisms or microbes. So that's why this is a triple corner process. Pollutants, what type of pollutants will be there, either inorganic or organic, and where that pollutants will be in the environment, either soil or water or air. So what type of organisms will be used. So this is the triple or triangle corner process. Now coming to the some of the terminology involved in the bioremediation before going to that one. Here is the xenobiotic. Xenobiotic means organic compounds generally not found in the environment. So like this will be include like insecticides, pesticides, herbicides, like petrochemicals, not petrochemicals, and the DDT example for that one. So these are coming the xenobiotic. So biomagnification means, in the previous lectures also we discussed that one. Biomagnification means accumulation or increase the concentration of the toxic compounds. Slowly in the organism tissues. That concentration will be increasing in the higher order of the food chain. So that is called the biomagnification or bioaccumulation. So best example taken the DDT. Dichloro-diphenyl trichloroethyl compound. This is a very good pesticide widely used for the agriculture. Mineralization means here the complete degradation. So whatever the pollutants that will be completely degradation to the either simpler carbon dioxide and water and other inorganic compounds. So that process is called the mineralization. In the bioremediation, the term mineralization means is a complete degradation of pollutants. Whatever pollutants will be either in water or soil or air, that will be completely degradation to the either carbon dioxide and water. Either partial degradation refers to the breakdown and intermediate stage compounds. Either complete degradation means final compounds are forming carbon dioxide and water. Is the partial degradation means intermediate state is formed. So bioconcentration factor means concentration of a pollutant from the environment. So how much concentration will be present in the environment? That pollutant concentration will be first take it, and the concentration in an organism compared with the environment. So that is the bioconcentration factor. So here what are the process will be involved in the bioremediation. So generally we are using the microbes or microorganisms we are using. Generally that will be releasing the enzymes. The enzymes are play a crucial role in the degradation or bioremediation of the pollutants. So these microbes will be release the enzymes to break down the contaminant or pollutant into the digestible pieces. So means whatever microorganisms we are using, that microorganisms will be release the enzymes. That enzymes break down the contaminants or pollutants to digestible small pieces. So now coming to the contaminants means what, for example, is the organic substances. So that is the digest as a food along with other energy source by the cell. For example, when the contaminants present, for example, here the contaminants will be there, we are adding to the some of the microbes or microorganisms to this one. The microorganisms or microbes also some energy will be releasing. So that energy plus whatever the organic substances during the digestion. So that energy will be also utilized by the cell, and the cell will be a degradation happen. So that is the bioremediation. So here is the main goal what is the main goal of the bioremediation means? So degradation of the organic substances. That hazardous living organisms are degradation by the organic contaminants into the inert products. For example, whatever is the hazardous pollutants, hazardous organic matter, organic substances present in that environment, that will be converted by using the living organisms degradation into the inert products, so not active products. So that is the major goal involved in the bioremediation process. So only simply says the harmless biological waste that all remain in the contaminants. So means finally converted to the harmless compounds. Harmless biological waste are converted to the hazardous waste. So that process will be involved in the bioremediation. So in this diagram also microbial cell also utilized in this one. Enzymes will be released, final product either carbon dioxide, water will be coming. So here the trapping, like this is a microorganism structure like this one. So once this oil is nothing but a higher fatty acid compound. So that is like a pollutant. When that will be coming enter into the whatever the cavity or microorganisms, that will be slowly by converted by the either cell activities or cell energy, that will be converted to the carbon dioxide and water. So that water will be coming out in this picture showing. So microorganisms digest oil and convert into carbon dioxide and water. So here the microorganisms give off and is sent to the out to the carbon dioxide and water. So this will be involved with the process in the bioremediation. Now coming to the what are the factors involved in the bioremediation? It's the factors. So the control and optimization of bioremediation process is a complex system, because is the why complex system means is the different types of pollutants, organic substances will be available in the different environments, either water, in soil or air. But that will be digested by the microorganisms, means that is not a simple system, a complex system. So many factors will be involved in the bioremediation. First one is the existence of microbial population. For example, here is the a pollutants will be there. In that we are adding to the microbes or microorganisms. So in that place, whatever that area, this microbe or microbial system or microbe system, that will be exist or not. That area or that place exist or not. That is the first. If not exist means these contaminants we cannot remove from that site. So means microbial whatever the population or growing the population must will be exist in that area, that place, then only the contaminants will be easily removed from the site. So second one, availability of contaminants to the microbial population. So when the population of the microbial will be increased, so the availability of contaminants will be there, then only that will be removed. Third one, so some of the environmental factors like a soil, type of soil, there are different types of soils are there. That soil, temperature, pH, presence of oxygen present in that soil, that area or that place. So these are all factors will be electron acceptors, nutrients for the growing of the microbial. So these are all factors are influencing the bioremediation in that place or that area. If the, for example, if some of the pH sensitive microbes there, so that place we cannot remove the contaminants. Is a temperature, some microbial not growing into the higher temperature conditions, so that will be not remove the contaminant site. So means these are all factors will be influencing the bioremediation. Now coming to the types of bioremediation, there are two types of bioremediation is there. First one is the aerobic bioremediation, second one is the anaerobic bioremediation. So aerobic means in the presence of the oxygen, anaerobic means in the absence of oxygen. So in these aerobic conditions, oxygen we are utilizing for the contaminants will be degradation. So that will be either involves utilization of oxygen to oxidation organic compounds. Whatever the organic compounds will be oxidized in the presence of the oxygen using. So these compounds may serve substrate for the supply of the carbon and energy to the microorganisms. For example, during the oxidation of the whatever organic compounds, some of the energy carbon will be liberated. That energy will be utilized for the source of the microorganisms. There are two types of enzyme generally used for the aerobic bioremediation: monooxygenases, whatever the this one aerobic conditions, and the second one is the dioxygenases are involved in the aerobic biodegradation. So means this reaction purely taken in the presence of the oxygen. Second one is the anaerobic bioremediation. Anaerobic means in the absence of the oxygen, bioremediation reaction will be completed. So anaerobic biodegradation does not require oxygen supply. So no need to require, in the absence of the oxygen, this reaction will be completed. So the growth of the anaerobic microorganisms and consequently degradation process are slow, because is oxygen will be not present in that one, means this anaerobic condition of the degradation process will be slow compared to the aerobic degradation. So some examples of organic compound degradator are listed below, given below. Hydrogenation. Hydrogen means a reduction of the whatever is the double bond compounds. So for benzoate, phenol, dehalogenation, PCBs, ethylenes, carboxylation, these are all compounds present in the organic matter that will be in the absence of will be degradation. So here is the what are the chemical reactions will be involved in the aerobic biodegradation and anaerobic biodegradation. For example, this is the organic matter or organic compound or organic substrate. So whence the in the presence of aerobic means in the presence of oxygen will be utilized. So when the oxygen will be utilized, so whatever the organic substances will be release the electrons that will be transferred to the oxygen. So means here the oxygen will be working as a electron acceptor. So means that will be working as a electron acceptor. Simply say the electron will be released the organic compound, that electrons will be taken or consumed by the oxygen. So means simply oxygen is a electron acceptor. So this immediately the organic matter will be converted into the carbon dioxide, water and biomass, and increased the number of bacteria also, because here is the microbial whatever is in the presence of oxygen. So that will be degradation. So second one is the anaerobic biodegradation. In this also whatever the organic matter, organic substances will be released the electrons, that will be nitrates are consuming, because we are not using the oxygen source. Anaerobic means in the absence of oxygen. So nitrates will be taken the electron acceptor, that will be converted to the end products like carbon dioxide, nitrogen and water, and biomass is obtained. So this is the chemical reactions will be involved in the aerobic biodegradation and anaerobic biodegradation. So means electrons will be taken by the oxygen in the aerobic conditions, is the nitrates will be taken in the electron acceptors in the anaerobic conditions. Now coming to the methods of bioremediation. So generally methods of bioremediation is simply classification of the bioremediation. There are two types of the bioremediation is there. One is the in situ bioremediation, second one is the ex situ bioremediation. Simply say in situ bioremediation means where the contaminants or pollutants in that site, that place, remediation will be solve that problem is called the in situ remediation. So means for example, one place some pollutants or contaminants is there, in that place only remediation will be or solve the using the microorganisms. Ex situ remediation means wherever the pollutants or substrates will be available in that area, the pollutants will be transferred or removed from that place and transferred to the some suitable places, reactors, labs, and then microbes and nutrients will be added to convert to the biodegradation process. That is called the ex situ bioremediation. So in situ means on-site process, ex situ means remove from that site. So that is the main difference between that one. Now we can go one by one. So in situ remediation means when the contaminants waste is treated at right as its point origin. So where the pollutants will be available or contaminants will be available, that place, that origin itself, treating will be takes place. So in that place is the whatever the microbes or microorganisms are will be added, and its site itself that contaminants will be removed. That is called the in situ bioremediation. So instead of removing and contaminant soil from the its place and not removing from that place, in situ bioremediation methods prevent the spreading of contamination during the displacement and transportation of contaminated. So in situ bioremediation method prevents is because we are not transporting that contaminants to the other place, other area. So means on site itself we are processing the bioremediation. That's why this will be not spreading contamination to the place. So in situ remediation further classification is there, intrinsic and engineered in situ bioremediation. So not that much going that one. Now coming to the what is the role of plants in situ bioremediation. Generally plants, microorganisms, whatever bacteria, fungus, different microbes will be utilized as a whatever source. So here is the plants. So plants, what is the role of in situ bioremediation? So here is the why different types of plants will be already trailed that one to be used as a phytoremediation. Some of the plants is discussed here: Bermuda grass, Bahia grass, roots, tall wheat. So these these grasses, whatever is the plant source, will be at the site, whatever is the contaminant site or polluted site will be there. So that plants containing the with high and toxicity tolerance and have excessive root binding systems. For example, in this place is a lot of whatever the pollutants or contaminants or heavy metals or toxicity is there. These plant roots will be more binding capacity of that pollutants or organic matter, easily absorb that pollutants. Then that pollutants will be absorbed and various forms of hydro compounds. So means once that will be absorbed means that will be completely removed on that site. So then next process, that process will be either phytoremediation, transformation or extraction, isolation. Next step will be taken. Here is the plants or different grasses will be utilized for the this role in the bioremediation. So different scientific names also, different grasses will be mentioned. Here is the Bermuda grass, sand and dactyline is a bahia grass, roots grass and a tall wheat grass. Now coming to the what are the role of microbes in situ remediation. So plants role, how plants absorb the toxic materials or organic contaminants or pollutants through the roots or binding capacity that will be using. Here is the microbes. Generally microbes is a breakdown the contaminants present in the environment. This is a source or food also, that one for that one. So this will be, the process is the anaerobic conditions and without oxygen when the microbes will be added, without the anaerobic conditions the products are methane, because is that there is no oxygen are utilizing. So final products also coming without oxygen compounds are producing. Is a methane is coming and hydrogen gas is coming, elemental sulfur is coming and dinitrogen gas is coming. In this all compounds end products are there is no oxygen, because in this process will be takes place in the anaerobic conditions without oxygen. Absence of oxygen conditions is the using the microorganisms using. So when it occurs, the presence of oxygen, so means in the presence of the aerobic conditions, the whatever the end products is related to the oxygen related compounds. Because oxygen we are using, that automatically converted to the oxygenated compounds only, carbon dioxide, water and related to some of the salts are coming. Here is the some of the microbes like bacteria, fungus and actinomycetes. Different actinomycetes are using as a microbes. So what are the advantages and disadvantages of the in situ bioremediation? So already this is the cost effective with minimal exposure to public and our site personnel, because we are not transporting that contaminants from the site. So means whatever contaminants is there, that place only is a remediation is giving. So that's why this is a cost effective. Not exposure to the public, because it's not transporting one place to other place. That what is not exposure to the public. So sites bioremediation remains minimum disturbed. So means is a whatever directly giving in that site, not disturb the any that area. So that's why minimally disturbed the bioremediation in this case. So what are the disadvantages means is the first one is the very time consuming process, because a lot of factors will be involving in this one. That's why is the time will be more consuming process, because in the site means whatever the environmental all conditions will be influencing on that pollutants. So either pH, temperature, humidity, these are all factors will be influencing. In that microbes will be either population doing or not. That's why it takes more time or consuming in situ bioremediation. So sites are directly exposed to environmental factors like a temperature and oxygen. That's why it takes more time. Microbial degrading ability varies seasonally. When the seasons will be changing, the same microbes also not degraded in that one in that place. That's why it's a seasonally change, because it's the environmental factors. Now coming to the whatever the ex situ bioremediation. Ex situ bioremediation means the waste or contaminants or toxic materials can be collected from the polluted sites. So means we are collected from that place, that area of the whatever waste, toxic materials or pollutant sites. That will be transported to the in the suitable places or reactors or labs. In that place, nutrients and water, these are all will be added and converted to the degradation. So here is the whatever is the pollutant, contamination, oil or soil will be collected from that one. is that there are different techniques will be utilizing and transported in the that one. Here is the slurry phase bioremediation, solid phase bioremediation. So these are different techniques will be utilized. So means is the contaminant site is the waste will be collected and transported to the suitable places like reactors, lab. In that place microorganisms and maintain the temperatures and degradation of the waste. So that process is called the ex situ bioremediation. So what are the advantages and disadvantages of the ex situ bioremediation? Means is this method is a better controlled and more efficient process because it's the completely taken whatever is the waste from that site and transport to the suitable place. So process can be improved by enrichment with the desired microorganisms. So for example, lot of environmental factors is not working in the laboratory conditions. So means this will be working efficiently and desired microorganisms you can directly use for in the reactors. Time required is very short because is that we are degradation in the laboratory means we maintain the temperature required or pH required or humidity required, whatever is the nutrients for the solutions required we are providing in the laboratory. So that's why the time consuming for the ex situ is less compared to the in situ process. So but this is the very costly expensive because is the why costly means is the waste or whatever the hazardous are transporting from one place to other place. So for that is a expensive process. So take a lot of necessary measures for the transporting that one. Sites pollution are highly disturbed because we are removing that pollutants from that side means site to whatever soil or whatever air that all related to surroundings also some of the disturbed. There are many disposable problems after process is complete. After completion of this process, a disposal problems also coming because is the degradation process, microbes, whatever is the final biomass also is a disposal problem is coming. Now coming to the kinds of remediation is the direct go to the strategies for the bioremediation. So there are different types of bioremediation process will be used in situ bioremediation and ex situ bioremediation. In the in situ, some of the strategies or techniques will be used for the control of the bioremediation, bioventing, biospurging, biostimulation, bioaugmentation. In ex situ bioremediation, land farming, compost, biofiles, bioreactor, phytoremediation. These are all different strategies or techniques are used for bioremediation in the in situ bioremediation and ex situ bioremediation. Now coming to the one by one simply discussed in this one. In the bioventing means is the simply supplying air and nutrients through wells contaminated to soil in indigenous bacteria. For example, in this diagram showing the whatever is the air will be pumping to the whatever is the nutrients to the wells. When the more air will be pumping means is the more degradation will be takes place. So that principle will be involved in the bioventing. Now coming to the biospurging. So biospurging means is the under the water table in the earth. So in that place is a very difficult to degradation take place is the contaminants. So because there is no oxygen supply. So here is the with the high pressure conditions the oxygen will be supplied. So means the process is called the biospurging. So means injection of air under the pressure below the water table. So below the water table increase groundwater oxygen concentration. So automatically oxygen concentration will be increasing. So then the rate of biological degradation of contaminants by naturally occurring bacteria will be increased. So means is the below water table we are increasing the oxygen concentration using the biospurging technique with the higher pressure conditions because is in that depth oxygen is not easily going that one you must apply the pressure. So now going to the biostimulation. Biostimulation is a simply say it involves the supplying oxygen and nutrients. For example, in this diagram, simply say this is the pollutant. A blue diagram is the all are pollutants. So this will be the microorganisms. A bacteria will be utilized. So for example, we added the more nutrients for the bacteria. So means bacteria will be population or growing that one. This whatever the pollutants in that site will be easily removed or degradation by this process. So means we are stimulating by using the some of the nutrients for growing of the microbes. So in the form of the aqua solutions. So that's why this process is called the biostimulation. So bioaugmentation means this is also another strategy or technique are used for that one. In this one, addition of the microorganisms indigenous and exogenous of the contaminant sites. So indigenous means whatever is the contaminants will be in that place or origin of that place. Exogenous means is the other place that will be coming contaminants. So in this one we are using more indigenous, whatever the contaminants will be easily biodegradation compared to the exogenous contaminants in this process. Now coming to the land farming, this is the very well known technique we are using daily in the agriculture field. So whatever the soil will be evacuated and periodically tilted by using the different techniques. Once the tilted of the soil means inside the whatever is the contaminants are there, that will be exposed to the more air. So once the more air means that will be easily degradation or degraded by the environmental conditions. So composting means, in this composting is a technique involves the combining the contaminated soil with a non-hazardous organic like a manure or agriculture waste. Once that will be added, the reach of the population of the whatever the microbes will be increased. That is the converted to the composting. Biofuels. This is the hybrid of the land farming and composting. So here is the essentially engineered cells are constructed to aerated composted files, means is the more oxygenated will be introduced in that one. Is the bioreactors. So generally in the ex-situ is one of the major important technique is the bioreactors. So here is the slurry bioreactors or aqueous reactors are used to the ex-situ treatment. Ex-situ treatment means, so whatever the contaminants will be collected from the site, that will be used in the different types of the reactors. So that soil or water pumped into the contaminated plumb. So bioremediation reactors involve the processing of contaminated solid or water through the engineered contaminated system. For example, here take the whatever is a reactor that contaminates such material and add the microbes and oxygen, pH maintain. These are all conditions that will be converted into the degradation process. So that is called the whatever bioreactors strategy. Now, phytoremediation. Phytoremediation means using the plants. Simply say using the plants. Plants have been commonly used for the bioremediation process called the phytoremediation. So, in which we are using the decontaminated soil or water and extracting heavy metals or contaminated from the whatever is the site. So, for example, here the roots are extracted contaminated heavy metals or organic matter will be easily absorbing the roots of the materials. So once that will be contaminated heavy metals, so break contaminated down the soils are contaminated. So usually plant will be harvested and removed from the site and burn it. For example, after completion of the whatever contaminants from that site, so you can harvest that plant by using the burning. So then some other process also is there, photovolatilization and phytoextraction. So means after removing the contaminants from the site, this plant whatever leaves and roots will be taken the extract. These compounds are entered into the extraction or not, you check through the different techniques. So that is the phytoremediation process. So this process is very effective in cleaning up the polluted soil. In the most polluted soil, this type of plant remediation widely using, because the plants are very good absorbent heavy metals, organic contaminants, and they're easily degradation or decomposed and absorbing through the roots like that one. So that process is called the bioremediation. So in this presentation we discussed about the bioremediation, in situ bioremediation, ex situ bioremediation, different strategies are using for the bioremediation, advantages and disadvantages of the different types of bioremediation. So in the next lecture we discussed about the solar energy. So thanking you.